Good morning.
Good morning, Abby. Hey, Patrick, I thought you can't make it today.
Our meeting got over quicker than I expected. So yes, I'm here. Happy to be.
See you again.
Same.
Right. So it seems like our drop-in area has calmed down. So let's get started. Welcome to Neuroccino on this Monday morning, 9:30. And today we have Eva presenting on Adaptive DBS for Parkinson. Will get FDA approval. We'll see about that in a minute. Just very briefly, the rules of the game. Everyone is welcome to present a paper. Just let us know, and then we discuss it to the best of our knowledge, and no presentation is needed. The only rule is it's not your own paper. Eva already shared the link in the chat. Thanks for that. So if you're ready, take it away.
Yeah, thank you. So the paper I'm presenting today is called Sensing Data and Methodology from the Adaptive DBS Algorithm for Personalized Therapy in Parkinson's Disease Clinical Trial. And this paper is actually from September, but it made the news recently because they got FDA approval as the first adaptive deep brain stimulation system for Parkinson's. So I just wanted to go a bit over the paper and first of all, some basic concepts. So deep brain stimulation is already used in patients in the continuous form to treat the cardinal motor symptoms of Parkinson's disease, as well as the ones that are induced by the medication. But it has seen limited effectiveness since patient symptoms are dynamic. So new technologies are trying to deliver stimulation where it's needed with directional leads, and also when it is needed with adaptive systems, such as the one in this trial. And in this trial, actually, they not only do the adaptive system, but in one of the cohorts, they use also directional stimulation. So this mix of directional and adaptive is the first report that there's been. So to adapt the simulation, they need a control signal, right? And what they use is a local field potential beta band oscillatory power measured in either the globus pallidus internus or the subthalamic nucleus. And this has seen relations with the fluctuation of symptoms in Parkinson and the responsiveness of medication and DBS therapy. And since it is actually present in 95% of patients off medication and also very high percentage when they are on medication, it is very applicable to most of the patients. So the ultimate goal is to minimize the need for medication, improve wearing off motor symptoms, since when you take medication, immediately you might have different symptoms than after a few hours. And yeah, reduce periods of dyskinesia from excessive therapy in DBS. So to dive into the methods section first, this is a global multi-center. I think 10 centers are involved across Europe, the US, and Canada, and mainly evaluating safety and effectiveness with 2 cohorts, as I already said, one with the directional stimulation as well. So the timeline for this study, they first, enroll patients and do this baseline phase, followed by a setup and an adjustment phase that is in the clinic, and it's just very in tune with the clinicians as well as the patients to make sure everything is safe and comfortable. And then it goes over to an evaluation phase based on which modes were actually successful in these set of phases. Sorry, my keyboard. As well as yeah, which ones actually work. So if only one worked, then they only do one mode. But if several worked, then they have this randomized design. And then they do a follow-up. And if patients really liked the adaptive algorithm, they can extend the use for the future. So what did they measure in this evaluation? The endpoints were mainly whether it works as well as the continuous form, right? So if on time, when the system is on, there is the same amount of lack of dyskinesia, basically. And then also the total energy delivered. So the goal is to have the same effects, but have to deliver less energy to the brain. They filled out a lot of questionnaires as well, on their symptoms, but also on their subjective experience to see which one they preferred. And they wore a watch for 6 days during each evaluation phase, so they could collect movement data from there. And the way that the system works is a closed loop, right? So we have this feedback coming from a sensor in the electrode that gets compared to a threshold that is previously set. And depending on this threshold, there will be an error. And with this, the controller would decide to increase, hold, or decrease stimulation. So this eventually acts on the electrode and the patient, and it will affect the physiological variables, which in turn gives us a new feedback variable. And it can also be disturbed by movement or medication. So this is continuously being evaluated. And there are, apart from 2 cohorts, there are 2 modes. So single and dual threshold adaptive DBS. For both modes, they made sure to set these limits to safe amplitudes, but also ones that would actually have like the therapeutic effects. So minimum threshold doesn't mean there is no stimulation. It just means it's the minimum to keep the symptoms at bay. And there is, of course, a lot of signal processing involved in this loop. So they are able to filter out movements and cardiac artifacts. And then for the dual threshold system, we'll get more into it later, but there is a slow adapting ramp. So it also filters out more of the movement artifacts because this is applied over a larger period of time. So the steps to actually program the algorithm are very personalized. So for each person, first you determine the stimulation limit without medication and then with medication. This is done for both modes, the dual threshold and the single threshold, since both will be moving in this band of stimulation. Then they determine the limit of the signal they're getting in. So the LFP signal, where do we place a threshold to either stimulate more or stimulate less, in the case of single threshold. And then for dual threshold, we need that band in which in the middle, it will just stay constant stimulation. But if we go over, we will get more stimulation and the other way around. Then there's also the possibility to optimize the ramp time. So for the dual threshold, this is a longer time, from 2.5 minutes to 5 minutes. And for the single threshold, it is a default of 250 milliseconds, but this can also be adjusted. So you can already tell that there's one more geared towards maybe fluctuation throughout the day. So what I was talking about, the medication wearing off, whereas the single threshold is more for very sudden changes. And then they also confirm that everything is good after medication wash in. Right, so going back up to the results. Yeah, they had 10 centers in the US, Europe, and Canada. And overall, they recruited 66 patients with different clinical subtypes and also different lead systems. So basically different hardware installed, and then different targets in different nuclei in the brain. And they were able to identify this signal in 84% of the patients overall. And medication didn't actually impact that much, being able to detect the LFPs, which means that this therapy can be implemented without needing to withdraw medication. And this number, they say, might be a bit inflated because some physicians already pre-screened participants with this peak presence. So it could be inflated, but actually this is within the bounds of what is already said in the literature, 75 to 95% of patients. And they also mentioned that most patients have a given type of leads, but that with the new SenseLight leads from Medtronic that are actually geared towards measuring these signals, there would be even better detection. So less contamination from external noise, such as cardiac artifacts. So a better visualization of this single and dual threshold. First of all, for the single threshold, we have that one LFP threshold. And when we go over it, then the stimulation should also be at the higher threshold of stimulation. And then when we spend time under the LFP threshold, the stimulation goes down to the minimum that was set. As I was saying, this ramp time is really high. 250 milliseconds. But it can be adjusted because this is actually where patients were most able to tell that they had adaptive DBS. So this ramp time can be adjusted so it's more comfortable. But overall, it's simple and it's fast. Then with the dual threshold, you see here we have 5-minute interval and the changes are way smaller. So in this case, if we are within the band, the stimulation will stay constant. If we are above the band, it will increment, but not go to the maximum. And if we are under, it will decrease, but not go to the minimum. So we don't have this trapezoidal shape that we see in the single threshold. And these patterns is not like one is better than the other. It's just, it allows for flexibility. So if a patient has uncomfortable motor fluctuations on the continuous form plus the medication, then maybe a dual threshold will work better because it works with the fluctuations of the effect of medication. Whereas if a patient was experiencing good control with the continuous DBS, but has challenges with narrow therapeutic windows of reduced efficacy, then with side effects such as in speech, then the single threshold could be a better fit. So this is the paper. They don't go into the final stats of which one they like better or not, because this was more at the start of the trial. But I can imagine it went well since they got the FDA approval, and perhaps a newer publication will come out shortly. But it's just a short summary of what they're doing and that it will actually get implemented in the clinic.
Awesome. Thank you very much. Super exciting paper. And I'm waiting because we have a question rule in this group. So I'm waiting for someone to volunteer who came late. I see smiles. Can I get some heads
will
up?
give Eduardo the stage.
Oh, that's very generous of you.
So kind of you.
Also, I missed like half of it, so I'm like,
Jesus. I just missed like a few seconds at the beginning, and now
Yeah,
I'm
it
not
wasn't
sure
half
about
bad.
the questions to ask. But yeah, So thank you for presenting. It's really a really cool paper. I was wondering, but again, maybe I lost the point at the beginning, whether they provide a little bit more detail on how they set up this adaptive feedback. So they collect from what I got, they collect the local field potentials, and if it's above or below a certain threshold, then the stimulation pops in or out. Is
Yeah.
that the entire point?
So for the single threshold, that is more or less, yeah, that is more or less it. So it is more of an on-off,
Yeah.
sorry. Yeah, an on-off system. And for the dual threshold, it's more incremental, but I don't know how big these steps are supposed to be.
Okay, because I've seen in figure 2 that they analyze like also like the power spectra of the signal. So I was wondering whether the stimulation was based also on like, I don't know, some power by frequency band or
So from what I understood, this figure is regarding the automated system to detect these peaks. So for different signals, this was the classification. So you can see that the one classified as high beta has the peak in the high beta range. But I think what they take is just when the peak is identified, they just look at the amplitude, and that becomes the what they compare with the threshold to determine where they are. So they're
Okay.
just taking the value of the amplitude of the LFP. That was my understanding.
So to identify patients better by
Sorry?
so it's basically a way to identify patients in a better way to like to provide a better stimulation for each patient, right?
I don't know what you mean by identifying patients better. This is
No,
like
I was just wondering what do they use this information for?
so the peak, the amplitude of the peak is what is then here essentially,
Okay.
and it's what is compared against the threshold to determine the stimulation that is needed.
Yeah, okay. Yeah.
Yeah.
Okay, thank you. I
You're
think
welcome.
Hannah has a question.
Well played.
Sure, but Lilith was first. I'm happy to go after Lilith. Since she raised her hand, it has to be fair.
Thank you, Eva, for presenting this paper. I have a question. So, this is how long should this therapy last? Did you mention this? For example, is it like continuously they have to come to clinic, or how long lasting is this effect? And the third question is the mechanism. Why does it actually treat the symptoms? And because Parkinson disease is mainly considered as far as I know, accumulation of some garbage in the brain that our system cannot flush it out. So while stimulating certain areas, does it somehow treat the cause, or does it only treat the symptom? And if it's only the symptom, then is it long lasting, these effects of this therapy? So yeah.
So I think this is in general for DBS, right? So first, patients are treated with medication and they try to avoid having the surgery. But by the end, the later stages of the disease, then it is already standard of care to, as far as my understanding goes, to have this surgery, get the electrodes, and then you will basically have them, yeah, the rest of your life. So it's
Okay.
you don't just need it for a period of time, but you need it, and it does get adjusted. So you do have to come into the clinic sometimes. They were working, or there are already systems out there where these adjustments don't need to be made in person. So you can have a clinician remotely adjusting your stimulation. But for this specific algorithm, I don't know if it needs a bit more adjustment or actually a bit less adjustment. But I know they also have control over it because they had an option, I don't know if it was an app, to opt out of this entirely if they had any period of being uncomfortable or they just wanted to go back to the continuous DBS, they could completely disable it themselves, the patient.
But can they do MRI scans after that or no?
I don't know.
Because no, I was just wondering, will the degeneration, the whole brain degeneration still appear? I don't know. Even though
I
you
also don't
have these no symptoms, so I just trying to understand the cause, if it's treating the cause or it's just helping to show that there is no problem, but actually the brain degenerates faster than a normal aging. I don't know, just not really specialized in this topic, so that's why you're asking.
Yeah, no, actually me neither, but I think it's more geared towards the symptoms. I don't know if there is any sort of feedback then to the actual cause mechanism by stimulating this way. I think it is, more geared towards the symptoms. So that's also why you want to minimize the dose because you're sort of doing a patch. You're not actually treating it. So the less energy you can deliver with the same results, the better. The less aggressive your patch on the symptoms is.
And so this FDA, that it is approved by FDA, so about the long-lasting effects. So at which point what was the last time point that they looked and say, yeah, okay, this is working well?
Yeah, that I don't know.
Or was it only like first month or first 1 week or one time shot? I don't know. Or they followed up for 1 year and they're, yeah, this is actually working for long term, that the device is not harmful, all these questions.
Probably to get this approval, I'm sure they need a long time. So even just the setup for the clinical trial, which I guess has to be completed to get it, is already requires 60 days here of adjustment, about 60 days for each, 30 days to evaluate each mode. Then long-term follow-up.
Here, yeah. 10 months.
So yeah, I can imagine this is basically like a year.
Okay.
Yeah.
Really cool.
But I think there will be an updated paper and trial. Yeah, it will just take longer than the FDA approval. Yeah.
But and the last question, so when we say that FDA has approved, does it mean that it passed all the clinical trials or?
I believe so. I believe it means that it can be.
So you can already use it for patients.
Yeah, that's my understanding.
Okay.
Yeah.
Cool, really cool. Thank you.
Eduardo has his hand up again.
Yeah, maybe just a short one. I don't want to steal time from Anna. So I just realized, so maybe is it possible they don't have a control group yet? Like they just have like a between like.
Yeah, I think when they mentioned the questionnaires, sorry, when they mentioned the questionnaires, actually they compare, I think, with their symptoms before. So in the continuous DBS, so yeah, basically this baseline acts as their control.
As
but
the control, yeah.
Yeah, they know they're getting this adaptive algorithm.
Yeah, it would be ideal to have maybe a
Yeah, and they need to know, so if they have any adverse effects, they can reverse back to the continuous.
Yeah, okay. Thanks.
Looks like Anna just briefly read the paper and is ready to bombard you with questions.
I did actually. I just read the discussion, and I was wondering. Thanks, Eva, for the presentation. I'm sorry that I missed a bit of it. I was wondering about like other implanted leads. So they speak in the discussion about that it's expected to be easier to detect those LFP peaks with patients that have implanted sense site leads. Do you know
Yeah.
anything about it? And, like, is it then, did they compare the two methodologies somehow, or is it like, what is the additional value of their work if there's already something like this out?
So both of them are Medtronic leads, as far
Right.
as I understand. It's just maybe they are newer ones, or they had a different design in mind. So I didn't look into it, but I imagine the first ones are maybe a bit older, and they were not expecting to need to record these peaks. And with the next ones, there is better noise suppression, and they're just better geared towards this. So whoever had the device implanted earlier, or maybe depending on what the different institutions were financing or not, maybe they have different leads.
Okay. And thank you. One additional question, tapping into the financing thing that actually Steph had and was trying to save me in the chat, giving me a question. Much appreciated. But it's about the availability and the openness of the system, basically. Is it open and like open source available for everyone, every clinician to use, or is it like linked to one private company who is becoming richer and richer by selling it to hospitals or to clinics?
So, I mean, this study was funded by Medtronic, and they are a private company. So my guess would be that they have, but all data analysis.
There is a quite long ethic declaration.
There is?
Yeah, I don't know, I just look for competing interest in the Control F.
Usually by default, it would be locked and patented to that particular company for the next decade. But I was just wondering if it's widely available, because if it is as impactful as it seems, then there would be great benefit if this was available.
But then you
The
couldn't
thing is
make
that
any money out of it. What do you mean?
So the thing is that they are already implementing in other places the adaptive system. So having this adaptive system is not new. It's more like the clinical trial is the newest thing, and actually evaluating the safety and putting in this protocol of how many adjustments it needs. So maybe it's not, I don't know if they have the patent for it necessarily, because this adaptive system has already been tested by other groups. So yeah, perhaps other people can implement it also with the information that they give here.
Cool. Valentina also has a question for you.
Hi, Eva. Sorry, I got in late, and maybe you already discussed this, but I was wondering whether you have the authors found any effects on the non-motor deficit impairment of the patients.
So yeah, the thing is that they don't get that far in in the paper. So you can see the results section actually starts here. It's just the detection of the signal, the automated algorithm to detect it, and this performance, but mostly based on the signal that they were able to extract. So they don't go into all the questionnaires that they measure and all the the effectiveness. So that's what I can imagine they've already tested and put through for the FDA approval. but that we don't have a publication on it yet. All the news outlet talking about this news, we're only referencing this paper.
I see.
Yeah. Yes. Hopefully we'll know more soon.
Thank you, Eva. And with that, we are right on time. It's 10 o'clock. So thanks a lot for joining us for Neuroccino this morning. And we'll see you again next Monday, 9:30. Have a great week in science. Bye.
Bye.